Good morning, good afternoon, or good evening to all of you that are joining us for our webinar today. I say that because we have students joining us from all over the world and all over the U.S. for our event today, from Sudan to Macedonia to India, Arizona in the United States, Florida. We feel truly fortunate to have such a wide array of individuals joining us from all over the world. We're excited to be able to offer this presentation in this format for you. And we're very thankful that we're joined today not only by Dr. Michael Peak, the program director for our biology and biotechnology programs here at William Paterson University, but we're also joined by several of our current students who are going to speak a bit about their research experience here at William Paterson. So exciting to be able to get some different perspectives on the program, to get to see what some of our current students are doing with their own research. And certainly for us, it's hopeful that this format is helpful for you, a way to sit back, take in some information about the program. Certainly it's our hope that if you are interested, we might see an application from you in the near future if you haven't applied already. And also too, would encourage you a little bit further down the line, if it's feasible for you, to consider visiting our campus. Beautiful campus in a wonderful location, just about 30 miles away from New York City. And for us, we have an open house coming out a little bit further out on August 18th. So want you to keep that in the back of your mind. Certainly it's a wonderful opportunity to meet Michael or others from his department, sit down, talk about your interest face-to-face a little bit further, like I mentioned, if that's feasible. We know for some of you there might be travel limitations involved, but certainly keep that in the back of your mind as well. One other thing I'd mention really quick, if time allows, we are going to have a question and answer session at the end of our presentation. But in the meantime, if you do have any questions that come to mind, if you do want something to be addressed by a member of our graduate admissions office, Michael, one of our students, feel free to type it in the chat box there as we go through. We'll either address and type an answer quickly or might read it off as a question at the end again if time allows. So lastly, I would let you know right under the handout section of the GoToWebinar screen, you'll see that this PowerPoint presentation is available in its entirety. So if you join us a bit late, if you have to step away early, there's the opportunity to go back and review what you might have missed. And similarly, keep in mind that a little bit further out, usually about a week or so, we are going to be sending this out as a link to the recording for this presentation up on our YouTube channel. So there'll be the opportunity again, whether you have a colleague, a friend, an associate who might benefit from taking part in this presentation, or if you have to leave early, there's the opportunity to catch up that way as well. So certainly that's more than enough from me. I'm sure you're here to hear from Michael and obviously our current students. So with that being said, I'm going to wrap up our intro and I'm going to pass it on over to Dr. Michael Peak. So take it away.
Well, thanks, Brian. I appreciate your time in putting all this together, and certainly appreciate everybody who signed in their time to listen to our programs and what we have to offer here at William Paterson University. So, like Brian said, we are about 30 miles west of New York City in a generally suburban area, highly densely populated. But as you can see, it's quite a picturesque campus. What you're looking at here on this first slide is our recently renovated science building, and so where we have a lot of state-of-the-art facilities. But along with campus, we have lots of other things in our area, like museums, hospitals, basic research with other research institutions in our area, as well as lots of research and development in the pharmaceutical and the biotechnology area. So there are lots of opportunities for you to explore while you're here, and many potential opportunities that you could possibly explore after you're finished here. So this webinar will consist of basically three things. I'll very briefly overview our programs and our curriculum. And what I wanted to highlight with this webinar is a focus of research. And what I did today is brought with you three current thesis research students that are going to tell you a little bit about their projects and give you a little bit of a different perspective about their experiences here, in the hopes that you would maybe potentially pursue a thesis research project. So with that said, I'll get started with our programs, our curriculum, and then I'll turn it over to our three research students. So we have two programs here at William Paterson University. They're both housed in the Department of Biology. Our first program is a 30-credit master's thesis or master's program in biology, and we offer two options. One is a thesis option and one is a non-thesis option. The thesis option is a research, unique, individually mentored research project that contributes potentially to a publishable outcome, whereas the non-thesis option is strictly coursework master's. The biotechnology is a 36-credit master's program, and again with both a thesis and a non-thesis option. So I'm going to highlight some of the program requirements that are required for each of these programs and hopefully distinguish between that thesis and non-thesis option. What those programs would potentially do for you, whether you choose a thesis or non-thesis option, is increase your research experience, obviously, if you do a thesis, but also we have some research opportunities for you if you did not do a thesis. But then we also have a lot of students that are interested in increasing their knowledge base. We have a fair number of students that want to pursue professional degrees, and so we have a lot of professional courses that might help increase your knowledge for an application to, say, medical school or dental school or some other such health-related program. And then we also have experience in giving people good preparation for wet lab techniques for workforce preparation. So with that said, the curriculum consists of a 30-credit biology master's program in which there are a few requirements for the thesis degree. We require everyone to take a course in research methods in biology, and basically that lays the foundation for a thesis student to do independent research. Thesis students also take 6 credits of thesis research, and they also take 1 seminar course in their area of interest, either molecular biology, organismal biology, or ecology. The non-thesis degree, like I said, is strictly a coursework master's. We do require students to be take that research methods course in biology, again, to provide them the basis for the foundation of biology, which is to do research. But then we also require them to take a seminar course, and then we do require, as a capstone experience for the non-thesis option, them to take some independent work. It doesn't have to be their own independent research, but it can be an independent reading where they do a literature review to demonstrate their knowledge, in-depth knowledge of a particular area. The rest of the coursework consists of electives, and so this may be a little small for everyone to see, but you can certainly visit the website link at the bottom of the page to look at all of our different elective courses. Again, you'll find a variety of courses in the molecular area, in the physiology area, and in the organismal biology area. For the biotechnology masters, it's a little more structured. There are 5 course requirements, which you can see there: molecular biology, DNA, cell culture, gene expression, proteins, and a seminar. The thesis option, again, exists for the biotechnology degree, in which you take 6 credits worth of independent mentored research. And then for the coursework, the non-thesis option, again, we require an independent project, whether that be an independent reading where you do a literature review, or where you can do a semester-long research project in the form of an independent study. The electives for the biotechnology masters are a little bit different than for the general biology degree, but nonetheless you can see from the list there what kind of courses are open to you. So I want to talk a little bit more about our research because the next part of the webinar will be the 3 students that we brought here to highlight their research projects. Our faculty consists of 20 faculty members, full-time tenured or tenure-track faculty members, and we have 3 areas of focus that our faculty can be grouped into. And the first area is ecology and evolution, and you see we have a list of about 7 faculty members that are involved in those sort of research areas. So we have a diverse array of people working in the field and in the laboratory, from Lyme disease and ticks to bat conservation down in the Pine Barrens, to coastal ecology systems, to even some local research projects here in a 100-acre preserve that we have adjacent to campus. The next group of research faculty that we have are in the area of physiology and behavior. And what you see here is we have organismal biologists that work on a diverse array of different topics, from organismal metamorphosis from Dr. Menon, looking at metamorphosis in tadpoles, to a very extensive vivarium that you see pictured here. So Drs. Benno, Lee, and Onivy, and now even Dr. Bierbauer work in our vivarium, where we have mice and rats that are used for both physiological and behavioral studies. And our last group of faculty is in the molecular biology and biotechnology area. We have about 10 faculty here working on a variety of different organisms, from bacteria to yeast to fungi. And what I want to say in summary about all of our faculty is that we're very active in some capacity, whether it be research or involving students in gaining access to research experiences. We have very extensive capabilities for ecological research, from field studies to field equipment. As I mentioned earlier, the physiology and behavior. We have an extensive or an advanced microscopy suite with both SEM and TEM and confocal. And then we have all the biotechnology tools to do all of your gel assays, et cetera, and PCR techniques. So with that said, we are a faculty that is very interested in mentoring students, both at the undergraduate and graduate level, and we have a series of 3 students here with me today to tell you about their research experiences in the thesis option in both the biology and the biotechnology programs. So first up is our student Casey Peterson, and he is working with Dr. Waldburger, and I'll turn it over to him right now.
Just like to thank Dr. Peak for the nice little introduction. Again, my name is Casey Peterson, and I work in Dr. Waldburger's lab. And a main focus of my study is on unique forms of gene expression in bacteria, particularly a bacteria known as Legionella pneumophila. So how I got started with my project is I did an independent graduate reading, which Dr. Peak had talked about as one of the requirements for the program, and I did a literature review of unique forms of gene expression in bacteria. And what I found is there's these unique proteins known as anti-sigma factors. And before I get into them, I just want to talk about sigma factors. So sigma factors are proteins that are needed for initiation of transcription. They bind to RNA polymerase, which then bind to promoters to turn on genes. Essentially, you need sigma factors to turn on genes. So when I was looking at this unique form of gene expression, these anti-sigma factors seem to be popping up, and anti-sigma factors actually sequester sigma factors and render them incapable of binding to promoters to turn on genes. So then I went and did a genomic database search and identified a potential anti-sigma factor in Legionella. So the potential anti-sigma factor was known as anti-sigma factor 28, and the sigma factor that this potential anti-sigma factor sequesters is known as sigma 28, which transcribes several vital genes of the flagellar regulon. So the transcription of these genes and production of flagellum are necessary for motility and infection. So essentially, if this anti-sigma factor proves to be a true anti-sigma factor, it will help regulate flagellum production in Legionella. So just a little bit about Legionella. Legionella has 2 distinct growth phases, a replicative phase, which features a non-motile bacteria with little toxicity, and then it moves into an infectious phase where is characterized by the production of a flagellum leading to high virulence in the host. So essentially what we know from this is that in the replicative phase there is no flagellum, but then it moves into a phase which there is a flagellum. So essentially we know that Legionella regulates flagellar synthesis in some way. And what we believe is that this anti-sigma factor is working during the replicative phase as to hinder the bacterium from growing a flagellum. So how are we going to test this hypothesis? Well, first of all, we're going to see if the anti-sigma factor is actually binding to the sigma factor. And you do this by what is known as a bacterial 2 hybrid assay. This is just a simple assay using molecular biology techniques to just see if our anti-sigma factor and sigma factor interact. This is kind of just the first test to see if our potential anti-sigma factor is working as a true anti-sigma factor. The next phase of the project is just going to be create knockouts of this gene. Essentially what we're going to do is we're going to knock out the gene that is necessary for producing the anti-sigma factor. And what our hypothesis is that when we knock out the anti-sigma factor, that during that replicative phase when there is no flagellum, now that there is no anti-sigma factor to regulate transcription, that during that replicative phase we are now going to get expression of a flagellum. And we're also going to do other various gene expression analysis, and hopefully with all these results we can come up with a good research plan and hopefully get good data to eventually pursue a publication in the field. So now I'm going to pass it on to Dan Lupo, who is doing research on Karenia brevis, and he can talk to you a little more about that. Thank you.
Hi, my name is Danny Lupo. I work under Dr. Emily Monroe, where I'm investigating the molecular mechanisms involved in Karenia brevis programmed cell death. Just as a little background, Karenia brevis is a unicellular dinoflagellate that forms near-annual harmful algal blooms in the Gulf of Mexico. These blooms are associated with many negative economic and ecological impacts, an economic loss to the U.S. of up to $82 million a year, massive fish kills and marine mammal mortalities, as well as harmful human health from ingesting toxic shellfish or inhaling aerosolized toxins when you're on the coast. Most of these impacts are due to Karenia's ability to produce a potent neurotoxin known as brevetoxin. My research that I'm currently working on now looks more at bloom dynamics. So Karenia typically blooms offshore in the oligotrophic waters or the nutrient-poor waters of the Gulf of Mexico on the southeastern coast of the U.S. Here, blooms will reach high cell concentrations that could exceed 10 to the 5th or 10 to the 6th cells per liter. Then just due to ocean currents or wind currents, blooms will migrate towards the coastline where they will lay dormant for several months, up to a year at a time. Then, without any molecular fingerprint, blooms will rapidly terminate, very quickly. And there's not a lot of research looking at both bloom initiation and bloom termination. Most of the research currently going on in Karenia is looking at brevetoxin biosynthesis, etc. So what I'm looking at is a few potential hypotheses that will lead to this bloom termination. Some research that has gone down in 2014 seems to implicate programmed cell death as a potential option for Karenia bloom termination. This study showed that during cell death, Karenia exhibits high levels of Cas-based activity. Cas-based activity has been coincided with a form of programmed cell death. This study aimed to identify a Cas-based enzyme in the currently available Transcriptome at the time, and they couldn't find a bona fide candidate for this Cas-based activity. Luckily, in 2014, the Marine Microbial Eukaryotic Transcriptome project was funded by the Moore Foundation, and this gave us a lot more sequence data on these eukaryotic algae, including dinoflagellates. This was huge for those studying dinoflagellates because dinoflagellates are known to have very large genomes, up to 40 times the size of the human genome in Karenia. So there wasn't a lot of sequence data available, but luckily the project funded by the Moore Foundation included Karenia in their transcriptome sequencing project. So I was able to take this library that was produced from this project, and I actually studied the Wilson strain of Karenia, and I have nearly 90,000 genes that I could create a blastable library of and look for potential Cas-based candidates in Karenia. So currently, I have 2 potential candidates for the caste-based activity that was shown in a previous study. And what I'm working on, because we don't have transformation procedures in dinoflagellates in Karinia, I'm actually taking these candidate genes and moving them into a vector that I can express in E. coli. From there, I'll take my transformants, grow them up, and I'm going to, from a protein lysate, I'm going to try to determine if these enzymes that I've put into E. coli now exhibit caste-based activity using a few different caste-based assays. E. coli actually works as a nice system for this because bacteria don't undergo programmed cell death, so cleavage of potential caste-based candidates will be due to the proteins that I actually transfect E. coli with. So just to summarize everything I just talked about, no gene has been linked to the caste-based activity observed in any protist. All varieties of protists have shown potential caste-based activity, including Karinia. So because of the large genomes in dinoflagellates, we see Karinia as a nice potential candidate to identify the first caste-based enzyme in any protist. So successful identification of PCD machinery will provide insight into the molecular mechanism of bloom termination and help us develop better bloom management strategies and predictive strategies to see how long blooms might last. So thank you. I'm going to give it off to Ian Drake, who works with Dr. Peak now.
Good morning, guys. My name's Ian Drake. I'm a thesis student here focusing my studies on plant biology. My advisor, Dr. Peak, and I study a special cool season grass, American beach grass. This is a coastal grass that you're probably familiar with if you've ever been to the beach here on the east coast of the United States. It's the one that dots the sand dunes in those fenced-off areas that say do not walk on the dunes. And this grass is important for many reasons, especially for its role as an ecosystem engineer, which builds and maintains sand dunes here on the east coast, and it's also a pioneer species, which means it grows in an environment that is uninhabitable to other plant species, and it ultimately enables the colonization of less stress-tolerant plant species. It's also particularly important to humans due to its widespread use by the United States government in building and restoring these sand dunes because of its unique ability to catch moving sand and bury itself while stabilizing the dunes with its extensive root system. These dunes act as natural storm barriers that not only protect coastal ecosystems, but also the billions of dollars of coastal infrastructure that line our coasts. Now, I'm particularly interested in a unique and influential relationship between beach grass and what's called a fungal endophyte. Endophyte meaning inside plant, and in this case referring to a fungal infection that persists throughout the lifespan of the host plant. These fungus grow in a kind of branched hyphal network that you can see here on the slide between the intercellular spaces of the plant cells, and they can confer all these cool benefits to the host plant like increased host resistance to herbivory, thermal stress tolerance, and altered photosynthetic capabilities. In particular, drought stress mitigation is particularly well documented, especially in agricultural grasses. And therefore, one of my overall research goals here at William Paterson is evaluating plant performance or beach grass performance in response to not only endophyte infection, but drought stress tolerance. And this is going to be particularly interesting, I think, because native or non-agricultural grasses are not well studied in regards to their relationship with these endophytes. So the first thing we did to start my research off was to obtain stock plants from the United States Department of Agriculture down in Cape May, New Jersey. Specifically, we obtained plants all of one commercial strain or genotype, what's called the Cape variety. And it's important because it's used almost exclusively in restoration plantings by the government. And it's also interesting because it's not only more vigorous relative to other native types, but it's been found in one study to be 100% infected by these fungal endophytes. This 100% infection status could be very influential in restoration areas and could have implications on the ecology of coastal areas. For example, in another study, the presence of endophyte infection showed to slow the rate of ecological succession in a grassland environment and even had suppressing effects on plant species and arthropod diversity. So after obtaining these plants, we transplanted them for growth in the greenhouse, and over the course of 6 to 8 months, I've been treating them with a special type of fungicide in order to acquire non-infected plants. So after getting infected and non-infected plants, we plan to compare their responses to a range of water availability regimes and drought conditions and assess their performance in these differing water availability regimes. And now a second research goal of my thesis is to determine the infection rates in restored and natural East Coast populations. So currently there's very limited data on the infection status of native coastal populations of beachgrass. From the very little data that exists, there seems to be highly variable rates of infection. And currently there is no data on the dynamics of infection status of restored populations. So what we've done so far in regards to this is take a census of infection of both commercial and native strains that have been maintained in the greenhouse. And this data suggested that beachgrass may actually be losing infection over time. And our current studies include a diagnosis of samples taken from multiple native and restored field sites over a period ranging from 2010 up until now in New Jersey. So that's what I'm doing. Thanks for listening, guys. I'm going to hand you back over to Dr. Michael Peak.
Well, I want to thank Casey, Dan, and Ian for their time and very eloquently explaining their research programs. And I think, underwritten in all of their talks is the close mentorship that they have with their faculty member to develop such good projects. And so hopefully these guys will be successful in their future careers. So with that said, I have two more things to leave you with, and that is how to get in. So we have fairly specific admission requirements, and basically that entails a bachelor's degree, preferably in the field of biology or some biologically related field. And what we're looking for with from the undergraduate institution is a good foundation in biology. So you should have courses in general biology, cellular biology, genetics, along with those co-requirements in chemistry and organic chemistry, physics and mathematics, particularly calculus. For the biotechnology program is a little bit more structured, that we have at least some good biochemistry background, but also some genetics background, and if you have any molecular genetics background, that would be also useful. So we accept applicants with a 3.0 on a 4.0 scale, and then the GRE is also a requirement, but that can be waived or substituted for the MCATs or the DATs as well. We also require 2 letters of reference, and those should be from individuals that can assess your academic ability. And then for all of you international students, you're required to have all of the above plus your WES translated marks as well as an English proficiency exam. And you can also see all these requirements on the link at the bottom of the slide. And then finally, we offer a couple different ways in which you can support yourself here at William-Paterson. We have a small number of graduate assistantships. So the 3 students that you heard from today are all supported under our graduate assistantship program. And so preference for those positions are given to those that are pursuing thesis research. And those are nice because they come with a small stipend, but also tuition remission. And so those are quite attractive, but they're also highly competitive. And an additional way that we can offer some financial support is that while you're pursuing your master's, you can also teach at the undergraduate level. So mainly what we try to do is we try to put our master's students in undergraduate laboratories, whether they be our general biology laboratories or some non-majors courses. So you can teach up to 6 credits of laboratories as an adjunct, and not only does that pay you, but it also gives you teaching experience and something to put on your CV or your resume. And you can find more information about that with the link. So with all of that, I want to thank you for listening and participating in this biology and biotechnology webinar. and I'll leave you with my virtual business card with all my contact information, and I will turn it back over to Brian and see if we have any questions.
Thank you very much, Michael. And to our grad students, that was wonderful and a great opportunity to see what some of our current students are doing with their research and the opportunities that our programs here can afford for students. If you do have any questions, as we referenced earlier, you can feel free to type them in the chat box there, and we'll make sure that we address them. The other thing, for anyone who may have joined us late, I just wanted to remind you that the PowerPoint presentation in its entirety is available under the Handouts tab on the GoToWebinar screen right there. You can download the PowerPoint, refer back to Michael's contact information, some of the links that he referenced. We're also going to be making this available to you in the near future on our YouTube channel, so there's the chance to either refer back, or if you have a colleague, a friend, or somebody else who wasn't able to join us today, the opportunity to share that link very easily so someone else can take part and review this webinar. And we did have, Michael, one question that came through. In terms of the length of the program for biology or biotechnology, for a typical student coming in and pursuing it full-time, approximately how long would it take a student to complete either program?
If a student's going full-time, taking, again, this is not like an undergraduate degree where you take 15 credits. Most students take about 6 credits a semester, so 2, possibly 3 courses a semester. It is theoretically possible to finish in about 1.5 years, but on average, most of our students finish in 2 years. With that said, we do have some part-time students that will work full-time and take classes in the evenings. Most of our graduate classes are in the evenings. We do offer other classes during the day, and those can take 3 or 4 years to finish, but then again, that's only taking maybe 1 or 2 courses a semester or a year. So on average, about 2 years, but it is possible to finish in I would say about 1.5 years.
And then for a student, a domestic student here in the U.S., if their GPA or their GREs or one part of the puzzle is coming in a little bit lower than what you listed in the requirements, do students have the opportunity to take coursework as a non-degree, a non-matriculated student? Is that something that you recommend to students, or is that something that you wouldn't advise for a student who is considering the program?
Yeah, let me first state that we look at the application as a whole. So if you have a GPA that's slightly less than the 3.0 threshold, we will review it, but that should be accompanied by very strong GRE scores and very strong letters of recommendation. If your application is reviewed and you're not accepted for matriculation, there is the potential for you to be a non-matriculated student, so basically a non-degree student, in which case if your GRE scores or your GPA isn't quite acceptable, you can take up to 3 courses here as a non-degree student to try to make your case to say, yes, I am have the adequate background and I am a good student, and bolster your case for admission to the program.
Thank you very much. And just to sort of build off of that a little bit, for anybody who's not aware, for domestic students, there's the opportunity to take up to 9 credits, which is typically 3 courses on the graduate level, or up to 30 undergraduate credits when you are not matriculated formally into a program. It gives you the chance to, for some students, sort of dip your toe in the water and see if grad coursework is the right fit, and for some it can be the opportunity to prove that you're ready and able to pursue and complete graduate work successfully, get the grades necessary, and for some students that's a way that they find themselves in a matriculated program after they've completed the 9 credits. So just something to keep in mind as well. And then to reference one other thing that Michael went over earlier on one of his slides, the course-by-course evaluation for any potential international applicants. It doesn't necessarily have to be from WES, it can be from any NACES approved credit evaluation service, so that's just something to keep in mind as well, but that is a requirement as a part of the application process for our international students. But it looks like that's it for our questions as of right now. I know we answered some before as we went through. On behalf of the Graduate Admissions Office, I want to thank all of you again for taking the time out of your schedule to join us. It's certainly our hope that this format was helpful for you and convenient as a way to learn more about the program. As I mentioned earlier, and I can't stress enough, if visiting the campus is a feasible option for you, and August 18th ends up working with your schedule, we'd love to have you come out, have the opportunity to meet Michael face-to-face, talk a bit more about your interests in the program, about research, etc. So certainly keep that day free if that's a possibility. I believe it's 5 to 7 p.m. that day. And otherwise, on behalf of everyone in the biology and biotech programs, for our current students, for Michael, myself, thank you so much for taking the time to come out. And Michael, thank you so much for the presentation. Certainly wonderful. And for our students, thank you so much for giving up your time. And it was certainly insightful to hear about some of the research that you're doing here on campus. So thank you, everyone, and enjoy the rest of your day.